Thank you so much, everyone. Thanks for the invitation to come here and share the opportunity to share what we've been working on our discovery of CDX-6512, GI stable methionine gamma lyase, also known as MGL, as a potentially orally administered enzyme therapy for HCU. So before I jump into the presentation, just a little bit about me. I'm currently a translational scientist and patient ambassador at Codexis. Codexis is an enzyme engineering company in the Bay Area of California. Codexis has been around for about 20 years, and we expanded into biotherapeutics about 9 years ago. Our first biotherapeutic candidate was for PKU. It's an orally administered therapy very similar to this. And that is now in clinical trial being managed by Nestlé Health Sciences. So I've been focused on therapeutic development for inborn errors of metabolism for about 20 years. The last 5 of those years have been at Codexis. I'm a patient advocate, and I'm very proud to say that many of the projects that I worked on while I was at the University of Pittsburgh before joining Codexis were funded in part by patient organizations. And just on a personal note, I am a dog lover and also a motorcyclist. So to jump into the science here, let's see if I can get this to advance. HCU is an inborn error of methionine metabolism characterized by mutations most commonly in the cystathionine beta synthase enzyme, or CBS. Methionine is an essential amino acid, meaning it's acquired from diet. So when CBS is defective or deficient, both methionine and homocysteine can become elevated. HCU is screened for at birth in all 50 states of the United States, but due to different testing protocols and different methionine cutoff values, it's estimated that about 50% of patients are missed at newborn screening. So the incidence numbers that are here up on the screen, HCU may be a lot more common than we currently think. The systems most commonly impacted by homocysteine toxicity is the brain and nervous system, and this can cause psychiatric and behavioral disorders, cognitive disabilities, movement disorders, and seizures. The skeletal system, which can cause osteoporosis and bone deformities, the cardiovascular system, which can cause abnormal blood clots or stroke, both of which can be life-threatening, and the eyes, which can manifest as lens dislocation and nearsightedness. The current standard of care basically leverages different parts of the methionine pathway. The only approved therapy is lifelong protein restriction. And so this manipulates the amount of methionine coming into the system via your diet. This special diet is often paired with different forms of supplementation to help remove homocysteine from the system. And this could be with vitamin B6 for B6 responsive patients. This is estimated to comprise about 50% of HCU patients, or with betaine, folate, and vitamin B12 that helps to recycle toxic homocysteine back into methionine. So the goal of an oral therapy would be to allow increased methionine tolerance, reduce severe consequences such as neurological symptoms and stroke, and improve quality of life. And at Codexis, we aim to do this with an enzyme substitution therapy. And this would be with an evolved bacterial methionine gamma lyase, or MGL, that can break down methionine coming in from diet at the site of protein breakdown, which would be the intestines. There we go. So considering the patient needs, we had a few goals in mind when developing this therapy. Oral administration with a meal would be non-invasive and available to both kids and adults. Since HCU is a genetic disorder and homocysteine can begin accumulating at birth, children are a very important patient group to include in our therapy. It needs to be highly specific for the amino acid of interest. For HCU, that would be methionine. And a decrease in methionine should also impact homocysteine levels. So this would reduce toxicity and allow for a relaxed diet, as well as increased protein tolerance. And the enzyme must be active in the GI tract so that it can remove methionine liberated from food prior to conversion to homocysteine and absorption into the bloodstream. So how did we go about doing this? Codexis has been involved in, we're an enzyme evolution company. We've been involved in this for a very long time, not always for biotherapeutics, and our system is the same as we move through evolution of these different enzymes. So we start with an enzyme with some of the characteristics that we want, and then we systematically evolve to improve what's lacking. We start by expressing libraries of variants in high throughput to assess different changes that we've introduced. Changes, these are essentially mutations, but mutations are not always bad, and I'll explain a little bit as I move forward. We then screen in simulated gastric and intestinal conditions, because these are the environments that these oral enzymes will be exposed to, and we want to make sure that they're stable and can function at the same state that they were before, or even better, to be a viable therapy. These variants are then ranked, and we typically have a pretty diverse spread of results. You can see here, all the green is an example of all the different variants that we would get out of a library. Some are a little bit better than where we started, which is the red bars. Some are worse, and then some are significantly better from where we started. We then take these, all of these variants, we sequence them, and we use artificial intelligence and machine learning to deconvolute the role of each change, which could be good, bad, or neutral. Once we understand where all these changes lie, what they're actually doing for the enzyme, we can take all of the beneficial changes from all of the different variants and roll them into our next round, into a single variant, and see if how that improves things moving forward. So we call this whole process a round of evolution, and this is repeated until we achieve our goals for a therapeutic candidate. For MGL, we went through 12 rounds of evolution, and over 27,000 variants were screened. And at the end of this process, whoops, that kind of jumped ahead a little bit. At the end of this 12-round process, we got what we are calling CDX-6512. And at each stage of this whole entire process, including the end, we screen for various biochemical characteristics. We assess activity, as well as activity when challenged in different environments, such as high temperature, low pH, exposure to proteases, such as pepsin, which is found in the stomach, trypsin and chymotrypsin, which is found in the intestine. And we also assess in simulated gastric and intestinal environments, which combines several of the above challenges into one screen. We found that 6512 was highly stable and active in these different in vitro screens, and then we moved forward into proof of concept studies in animal models. So in these studies, we fed a high methionine protein source and then treated with MGL to see an effective treatment. And we focused on two animal models, the I278T HCU mouse model and healthy non-human primates. And I'll give a little bit of background about each of these models as I move forward. But first, I want to talk a little bit about a window to see an effective therapy. This is an important part of any study. For these studies, we used two very different protein sources, and which one we used was dependent on the response window that we could generate in the animal model. Whey is a natural protein found in milk, and it's a very relevant dietary protein source because it's something that people eat normally. It's a full-length protein. There's no free amino acids. And on the other hand, peptone is a non-natural protein source. The peptone that we were using, the base protein is casein, which is a natural protein found in milk also, similar to whey. But the peptone, to make the peptone, the casein is pre-digested with enzymes to chop it up into small pieces. So it's actually mostly free amino acids and some peptides. So what do I mean by a response window? For the HCU mouse, and I don't have access to a cursor, so I'll try to talk through this. For the HCU mouse, they're normally maintained on standard protein diet, and their serum homocysteine levels when on standard diet, shown by the black bar, are very high. And this is essentially the maximum amount that is seen in this animal model. What's common to do for any sort of feeding study is to fast before the study is started. And so you can see with an overnight fast, the serum levels do drop for homocysteine, seen by the white bar. And then with whey protein challenge, it then increases slightly again. So this difference between the overnight fast and the whey protein, this is our response window. And in general, the larger the window we can generate, the better chance we have to see an effective treatment. This response window here is rather small, especially when you're talking about animal studies where there's always going to be a variability between the individual animals in your study. So at this point, our collaborator, Warren Kruger, suggested that we pre-treat with low methionine diet to further reduce the homocysteine levels and essentially prime the system to respond to the whey. And you can see that our response window with low methionine diet and then following the whey protein is much larger. So this was what we decided to move forward with in these studies. And what's also important to note is that by doing this, we're able to continue to use whey, which is a big benefit because this is the most relevant protein source. For healthy non-human primate, on the other hand, we wanted to use whey protein if possible, of course, because this is the most relevant protein source, but we're contending with a healthy methionine pathway. And methionine metabolism is a very efficient process. Also note that for non-human primates, we are focused on methionine, not homocysteine like in the mouse, because again, these are healthy animals, and so there isn't an opportunity to look for changes in homocysteine. We found that in the monkey, the best window was obtained with peptone. So then moving forward into our HCU mouse designs, just a little bit of background about these different models. So for any animal model, there are pros and cons. The I278T mouse is a model with a human HCU causing mutation, and it's a very good model of human HCU. It has a lot of the characteristics seen in humans: elevated methionine and homocysteine, improvement with low methionine diet, some of the other downstream effects. And it also does not have early neonatal lethality like the CBS knockout mouse model does. However, mice are very small with a very fast metabolism, so there are limitations. Transit through the GI is very, very fast, not at all like human, and we also can't give very much, we can't dose very much or take very much blood after just because of their size and the limitations there are. There's also the issue with the response window, but we did sort that out with our low methionine diet. So we have a much better response window with low methionine diet than using whey protein. And so what we did was pre-treat with the low methionine diet for 7 days. We then treated with whey containing methionine with and without MGL. And then we took a series of blood samples to see how that was impacting their serum levels. And we took one blood sample prior to the weigh meal to assess the baseline, and then 3 different time points out to 24 hours in this first study. There we go. So in this first study, we treated with 148 milligrams per kilogram of CDX-6512 or vehicle following the whey protein meal. Homocysteine was increased at all time points. So this is shown by the gray bars, the difference from low methionine diet, which is our baseline, seen by the white bar. And homocysteine was suppressed also at all time points, shown by the blue bars. This suppression was statistically significant at 45 minutes and 4 hours, and that corresponded to 28% suppression of serum homocysteine at 45 minutes or 31% at 4 hours. You can see that methionine was also increased with the whey protein and suppressed with MGL. One thing I do want to note, want you to notice is the difference in scale here for homocysteine and methionine. We are getting a lot more. There's a lot more homocysteine in the serum than there is methionine. Also note that the methionine and homocysteine peaked at different time points. The highest level was seen at 4 hours for homocysteine, but for 45 minutes at methionine. However, since at 4 hours it was statistically significant for both of these measures, that was the time point we decided to move forward with in our follow-up study in the mouse model. And so in this follow-up study, we did a dose response down to 37 milligrams per kilogram. And for serum homocysteine, we saw a dose responsive effect with greater impact in the homocysteine occurring with the higher dosing of MGL, up to 45% homocysteine suppression compared to the whey protein alone, seen by the gray bar. And despite the fact that methionine was not statistically significant in this particular study, it may be hard to see, but MGL was able to suppress the average serum methionine to levels below pre-dose baseline. The baseline is indicated by the dotted line, and in all of our dosing, it is slightly below that level. This amount of suppression in methionine with the highest dose, 148 milligrams per kilogram, is actually the same effect we saw in our prior study at 4 hours at this dose level. So we had very good agreement between these two studies. So then moving on to our non-human primate studies, there are some benefits to using monkeys. One obvious one is that they're larger. The transit time through the GI is much closer to human than for a mouse. Monkey is also much better for estimating human dose for clinical trials. And because they're larger, we're less limited by how much we can dose, and also the number of samples we can collect during the course of the study. However, as mentioned before, these are healthy animals. We're contending with a methionine pathway that is intact, and there's no opportunity to see an impact on homocysteine. We're also unable to use whey protein for this, and instead used peptone to generate a response window for methionine. The general study design was actually very similar to what we did for mouse. We fasted these animals overnight, and then peptone was given with or without MGL. And then blood was collected over 24 hours. We were able to collect 2 different time points pre-meal to get an accurate baseline, and then a lot more samples after to get a nice response curve over the entire time course. And I realize this slide is a little bit more busy than the others, but if you just look at the left graph first, the time course data, what we did was a dose response down to 37 milligrams per kilogram. With peptone alone, shown by the black line, you can see a very high spike that peaks at 2 hours and then comes back to baseline by 8 hours. I'm not showing the entire 24-hour time course because baseline, it does return to baseline by 8 hours. And then with MGL, with the different doses, you can see a very prominent effect in a dose-responsive manner in methionine suppression. And this corresponds to suppression up to 70% at our highest dose. If you look at the right graph. What seems to be happening here is we're seeing maximal effect at doses 92.5 milligrams per kilogram and above. All of those doses are very highly significant, 64% to 70% methionine suppression. And again, these are healthy animals, so we didn't assess homocysteine. So we are in the progress of assessing doses lower than 37 milligrams per kilogram to better determine possible daily dose, as pill burden is something we are very conscious of for any oral therapy that we're developing. And also note that all the studies that we've done so far have been just a single dose, looking at efficacy after one single dose of MGL, and that's not how these therapies will be intended. This will be a daily therapy that you would take with a meal. So it's possible that repeat dosing may also reduce the amount of material you would need to take on a daily basis. So those studies are in progress. So in summary, a bacterial MGL enzyme was engineered as an oral therapeutic with high GI stability and activity in the small intestine to degrade methionine liberated from food. In the HCU mouse model, a single dose suppressed serum homocysteine in a dose-dependent manner up to 45% following a whey protein challenge. And in healthy non-human primate, a single dose suppressed plasma methionine in a dose-dependent manner up to 64% following a peptone challenge. So this work highlights the prospect of a GI-stable amino acid degrading enzyme as a potential therapy for HCU. Codexis has also applied the strategy to target other inborn errors, such as PKU, which is now in clinical trial, and MSUD, which is still in preclinical development. So I'd like to thank everyone who's been involved in this project at Codexis. This has been in our pipeline for a while, and there's been a lot of people that have been instrumental to getting it to where it is today. We are continuing through development and are aiming towards, we're looking forward to getting this into clinical trial. I'd also like to thank our collaborators at Fox Chase Cancer Center in Philadelphia who performed the HCU mouse studies that were summarized here. They've been a big help in helping to talk through a lot of different aspects of our therapy and how to design our mouse studies. So thank you so much for all of your help. And thank you, and I will take any questions. Thanks so much.
Thanks very much, Kristen. Let's see if we've got any questions from the audience. We'll give it a minute or two. It's great to hear there's so much going on in terms of research in the homocysteine areas. Okay. Well, in the absence of any questions, we'll say a big thank you for your presentation. We really appreciate you sharing the research activities that are happening at Codexis. And
yep, thank you so much.
We'll